So my dear viewers, you are all warmly welcome to our Vidhyalakshmi YouTube channel. I am Mrs. Lata from Botany department. So today we are going to see the topic, that is the embryo sac development. Okay. Already we discussed, that is the previous topic of the embryo sac development, that is a megasporogenesis. As a result of megasporogenesis, the megaspore is formed, isn't it? Now the megaspore is undergoing the division to form an embryo sac. Okay. So that is we are going to hear. So based on the number of megaspores that is develops into the embryo sac. We know very well the megaspores develop into the embryo sac. As a result of megasporogenesis, 4 megaspores are formed. Okay. So under the 4 megaspores, so any one of the megaspore will develop into the embryo sac, that is known as a monosporic in conditions. Mono means that is a single, isn't it? Okay. So one of the megaspore will develop into the embryo sac, that is a female gametophyte, that is known as a monosporic. So example for the monosporic, that is a polygonum. Okay, monosporic development, that is example is a polygonum. And what is the bisporic in conditions? So among the 4 megaspores, only 2 megaspores are involved in the embryo sac development, that is known as a bisporic in condition. Can you give the example for the bisporic? That is a allium. Okay, the name of the species is allium. And third one is here tetrasporic in conditions. So what is the meaning of tetrasporic? All the 4 megaspores are involved in the embryo sac development is known as here tetrasporic. Okay. So example, that is a peperomia. Okay. So we are going to see one by one. The first we are going to see the monosporic in condition. So monosporic is otherwise called, that is a polygonum in type. Okay. So of the 4 megaspores are formed, already we studied, there is a 4 megaspores are formed. So only 1 megaspore is act as a functional megaspore. So remaining 3 megaspores are degenerated. Okay. The functional megaspore is the first cell of the embryo sac or female gametophyte. Okay. So what is a polygonum type and enothera type? Polygonum type means the embryo sac is developed from the chalacal megaspore. Okay, the megaspore is towards the chalasa end. Okay, enothera type means the embryo sac is developed from the micropylar megaspore. The megaspore is situated towards the micropylar end. So that is known as a enothera type. So one is a polygonum type, another one is the enothera type. So now we have to see the polygonum type. Okay, we can see the polygonum type in the monosporic development. So this is the development of monosporic embryo sac, monosporic development. So in the development of ovule and embryo sac, that is known as a polygonum type. Okay. So already we know that is a nucellus. Nucellus means that is a mass of continuous mass of the parenchymatous cells present in the ovule. Okay. So from the nucellus, a hypodermal cells are organized. Okay. The hypodermal cell which gives rise to the archisporial cells. That is act as a archisporial cell which gives rise to, that is the hypodermal. Archisporial cells are undergoing the divisions to form a 2 type of layer, that is we already studied in the megasporogenesis. Okay. So finally megaspore mother cells are formed. The megaspore mother cells are undergoing the reduction division to form a 4 haploid megaspores. Okay. The 4 haploid megaspores, that is 3 megaspores are degenerated. You can see here the 3 megaspores are very smaller in size, that is a degenerated in condition. This is degenerated. Okay. So the only one is the functional megaspore. So from the functional megaspore, the embryo sac is formed. Okay. The first functional megaspore is act as a first cell of the female gametophyte, or that is the embryo sac. Okay. So that is undergoing the divisions. The first functional megaspore is a nucleus that is undergoing the divisions. Okay. So what type of division is occur? So that is a mitotic divisions. As a result of mitotic division, which gives rise to the 2 daughter nuclei. Okay. So 2 daughter nuclei is formed. Initially the functional megaspores, they have to elongate their size along with the micropylar axis and the chalacal axis. Okay. When it reaches a certain size, okay, or certain size vandadukku appuram da adu vandu division agum. The first division is a mitotic division. As a result of first mitotic division, 2 nuclei is formed. Okay. The 2 nuclei is, that is moving towards the opposite pole. Okay. So how can they move? So with the help of the vacuole. So where is the vacuole? The vacuole is present in the center of the nuclei. Okay. So the vacuole is expand and push the the polar nuclei, that is both daughter nuclei will move towards the polar end. Okay, that's a polar means that is a micropolar end and another one is a chalosal end. The both of them is moving towards the micropolar and chalosal end. Okay, then second mitotic division will occur. So as a result of second mitotic divisions, the 4 nuclei is formed. Okay, so the 4 nuclei is present here. The 4 nuclei is again undergoing the third mitotic divisions. Okay, as a result of third mitotic divisions, 8 nuclei is formed. That 8 nuclei is, that's a free nuclei in the cytoplasm. So very important thing, the cell wall formations does not occur follow the nuclear divisions. Okay, so all the cells should be formed. Finally the cell wall formation should occur here. Okay, all the 8 nuclei are free in the cytoplasm. Okay, the 8 nuclei, so that is followed by the cellular organizations of the embryo sac is happened. Okay, so what is the meaning of the cellular organizations? So here the 8 nuclei is, so they have to arranged by themselves. Okay, so actually 4 nuclei in the one end, another 4 nuclei in the chalosal end. Okay, now of the 4 nucleus, the 3 nucleus are together to forming a, that's a egg apparatus. Okay, so we can see the egg apparatus towards the micropolar end. Okay, so towards the micropolar end, the egg apparatus are present. The 3 nuclei is together to forming the egg apparatus. The 4th one is, that is a free, that's a left free in the cytoplasm. Here the 4th one will be free. Okay, so the 3 together to forming a egg apparatus. The egg apparatus consists of 2 synergies, that is a 2 synergies are present and center cell is considered as a egg cell. Okay, so one egg cell and 2 synergies are together to forming a egg apparatus. Okay, and the 4th nuclei is set free in the cytoplasms. Okay, the 4th nucleus that is considered as a upper polar nucleus, that is known as a upper polar nuclei. So the nuclei from the micropolar end is considered as a upper polar nucleus. Okay, and similarly the antipodal consists of the 4 nuclei. Okay, in the antipodals the 3 nuclei are together to forming a antipodals. Okay, and one of the nuclei is free, that is also free and in the cytoplasm. Okay, that is considered as a lower polar nucleus, that is considered as a lower polar nucleus. Okay, upper polar nucleus from the micropolar end and the lower polar nucleus from the chalosal end. Okay, the both nuclei is together to forming a secondary nuclei. Okay, that is known as a secondary nuclei or polar nuclei. Okay, so here all the nucleus are, there's a free in the conditions. Okay, they don't have any cell wall formations. Okay, so initially when the 8 nucleus are free, initially it is 8 nucleus are free in the conditions. So after that there is a cell wall formation should be occur. Okay, so how many cells are there? It has a 7 celled in stages. It has a 7 cell, but 8 nuclei is present. Okay, and each nuclei consists of a separate cells are there. Okay, so here in the synergies it has a 3 cells are present and the antipodals 3 cells are present. But what about in the center of the polar nuclei? They have a single cell. Okay, but it has a 2 nuclei. So that is known as a polar nucleus. Okay, so we know very well that after the fertilizations the polar nuclei so will change into, so that is your endosperm. The polar nuclei is fertilized by the second male gametes to form a endosperm, isn't it? So that egg is undergoing the fertilization to form a zygote. Okay, so this condition is known as the polygonum type. Okay, polygonum type means it has a 7 celled stages and 8 nuclei is present. So you can see the one more picture. So this is a 4 haploid nuclei is present, 4 haploid megaspores are there. So that's only one of the megaspore is undergoing that functions, remaining 3 are, so that is your degenerated. Okay, so that one is undergoing the first mitotic and second mitotic and third mitotic. Totally 3 mitotic division is occurred to form a 8 nuclei. Okay, the 8 nuclei is arranged in the embryo sac. That type is known as a polygonum type. So that next we have to see the synergies and antipodals, egg cells. Okay, so what are the arrangement of the synergies and egg cell and antipodals? Synergid cells. The synergid cells are containing the 2 number. How many synergid cells are present? It has here 2 numbers. Okay, ma? So that is present in the protoplasm with the nuclei. It has here, so that is a living substance are present. That is known as a protoplasm. And it has a vacuole is also present. Okay, very important structure is present here. So that is a special cellular thickening. Okay, a special cellular thickening is known as a filiform apparatus. Okay, the special cellular thickening, that is your filiform apparatus are there. Okay, so what is the function of the filiform apparatus? They have to absorb the nutrients from the new cells. Actually, it consists of your new cells. Okay, so new cells, from the new cells they have to absorbing the nutrients. Okay, they have to absorb and conduct the nutrients from the new cells to the embryo sac. So that is the importance of the filiform apparatus. Okay, and another functions of the filiform apparatus, they have to guide the pollen tube to enter the microscope. Actually, the pollen tube should be coming from the male gametophyte. Okay, that is the pollen tube. The pollen tube is enter into the embryo sac. Okay, so on the embryo sac varadukku the guide pannudhu, that is the filiform apparatus. And what is the functions of synergids? Okay, actually the synergids are degenerated. So after the fertilization, the synergids antipodals are degenerated. Okay, the synergids are secreting the some chemotropic substance. The chemotropic substance are attract the pollen tube to enter the embryo sac. They have to attract the pollen tube. And the substance அப்படியே அதை attract பண்ணி உள்ள கொண்டு வரும். அதே filiform apparatus வந்து they have to guide the pollen tube to enter the embryo sac. Okay. And only synergids are degenerated and develop the haustoria. In some of the species, the synergids are that act as a haustoria. Haustoria means that is they have to sucking the food material from some of the species. Yeah, sucking the food material from the host, that is known as a haustoria. It's a special type of the root. So next one is a XII, and either side of the synergids we have to see the XII. Okay, it is the only one XII between the synergids. The XII is also the polarized in conditions. Okay, it has a vacuole and the thick wall. Okay, it is covered with a thick wall which is towards the micropylar end. Actually, egg apparatus is towards the micropylar end. Okay, so that is a new cell is, the nucleus is chelacal end. That's a micropylar end and chelacal end. Vacuole is towards the micropylar end and nucleus is towards the chelacal end. Okay, so what is the functions of that is a egg? Actually, egg is fuses with the first male gamete to form a zygote. Okay, the zygote is the first cell, isn't it? The zygote will develop into that is a embryo. So the development of the monosporic, the last one, central cell. Okay, what is in the central cell? The central cell is consists of a polar nuclei. It has a 2 polar nuclei. The 2 polar nuclei is connected with the antipodals and egg apparatus. Okay, so that is a 2 polar nuclei. Before fertilization, we consider as a diploid in conditions. That's a 2n அப்படின்னு சொல்றோம். After the fertilization, that is your endosperm. Okay, after the triple fusions, actually the second male gamete is fuses with the, that's a polar nuclei to form a triple fusion. That's a 3n. 3n means that is a endosperm is formed. So what is the functions of endosperm? The endosperm will supplying the nutrition to the developing embryo. And antipodals. Okay, the antipodals are towards the chelacal end. So it has a protoplasm, that's a living substance, and starch, protein, lipids, and ascorbic acids and the oxidases, the enzymes also present here. So what is the main functions of the antipodal cells? They transfer the growth hormones and nutrients to the plant body. Okay, they have to absorbing the growth hormones and nutrients to supply to the growing embryo sac. Okay, so that's the antipodals. And next we are going to see the bisphoric in conditions. We studied the monosphoric in condition. Monosphoric na ஒரே ஒரு megaspore மட்டும் embryo sac developmentல involve ஆகுது இல்லையா? Bisphoric அப்படினா, so among the 4 megaspores, 2 megaspores are involved in the embryo sac development. Okay, so here the formations of the bisphoric in condition, example is the allium. Okay, so it is a allium type and endymion type. So 2 types are there. So the allium type is a chelacal dyad. It consists of 2 nuclei there. Okay, endymion type is a micropylar dyad. It's towards the micropylar end. It consists of, the dyad means it consists of 2 nuclei. Okay, so dyad means 2 nuclei. So next one is a tetrasporic in condition. Tetrasporic means all the megaspores are involving in the embryo sac formation. That is known as a tetrasporic in conditions. Okay, it has so many types are there. Adoxa type. Adoxa type means it has 8 nuclei. Similarly, polygonum. So 8 nuclei, only one mitotic division is occur. Actually, in the polygonum, 3 mitotic division is occur. As a result of 3 mitotic division, 8 nuclei is formed. But it has only the one mitotic division is occur. And next one is the plumpago type. Okay, plumpago type is the absence of synergy. It's a very important characters we mentioned here. It's everything is very important for the competitive examinations. So where the absence of synergies, that's in the plumpago type. And the antipodals. Okay, synergies and antipodals also absent here. Okay, next is pinia type. The 4 polar nuclei and the no antipodals. Okay, 4 polar nuclei. It consists of the 4 nuclei. Polar nuclei usually in the polygonum type it has only 2 cells are there. Okay, but here we can see the 4 cells and no antipodals. Okay, antipodals are absent. And tetrasporic like peperomia type. Okay, drusa type. Peperomia is egg apparatus which have only one synergids. Usually 2 synergids are present in the polygonum, but here only one synergids are there. Drusa type, large numbers of antipodals are present. And fritillaria type, triploid antipodal nuclei and tetraploid polar nuclei. Okay, so triploid it consists of 3n. Okay, tetraploid means it consists of 4n. Number of chromosomes is a triploid, number of chromosomes is a tetraploid. 4 chromosomes are present. So next one is a plumbicula type, that is a single triploid antipodals, no synergids and single cell egg apparatus. Okay, single triploid antipodal. This is a common type only. No synergids. Synergids are absent. Okay, and another one that's a single cell egg apparatus. Egg apparatus is only one. They don't have any synergids. Okay, egg apparatus alone, egg cells alone is present. And last one we are going to see the very interesting topic. That is a pollen embryo sac. Actually, we know very well embryo sac means that is the female gametophyte. Okay, pollen is the male gamete. Pollen is the male gametophyte, isn't it? So pollen embryo sac. Eppadi pollen engiradhu or male part, but eppadi embryo sac nu adha solrom appdin pathina. In some cases, the pollen grains usually two nuclei is present. Okay, the 2 male gametes formed in the male gametophyte. Okay, but here and some of the number of gametes will be, number of nuclei will be increased here. Okay, the pollen grain, the number of nuclei may increase. Okay, so number of nuclei should be increases. It's look like the embryo sac. Eppadi or 8 cell stage ah irukudho adhe mari nariya serumbodhu vandu it's look like the embryo sac. So then only we have to call this as a pollen embryo sac. Okay, so this type we can see in the some of the members of the Liliaceae. Okay, Liliaceae family, that's a resemble to the embryo sac. Liliaceae, all the Liliaceae, allium type அப்படினு சொன்னா Liliaceae தான். ஓகேவா, இந்த Liliaceae we could see the a number of nuclei. Okay, so number of nuclei should be that is a enormous nuclei that's a look like the embryo sac. Okay, so that is known as a pollen embryo sac. There is a new there is a very important information to you. Okay, pollen embryo sac, just we have to think about it. இல்லையா, pollen na male, embryo sac na female. எப்படி pollen embryo sac னு சொல்றோம் அப்படினு. So such a pollen embryo sacs were first reported in the Hyacinthus orientalis அப்படிங்கிற species, Hyacinthus orientalis by the Namak. Okay, the Namak is given so many classifications. In 1898, the scientist Namak is found the number of nuclei, so in the species Liliaceae. The pollen embryo sac is also called as a Namak phenomenon. Okay, because that is discovered by the Namak. Okay, so today I hope you could understand the topic. Just you have to recall once again so what we studied today. So the development of monosporic means only one megaspores are involved. Okay, thank you children. I hope you could understand. Thank you so much for watching this video.